In lecture, you are learning about pathogenic bacteria, some of which have a coccus shape. This lab is intended to complement those lectures by introducing you to some procedures or tests, also known as assays, that can help you distinguish between various coccibacteria, many of which are pathogenic. Before going any farther, it is highly recommended that you read through the introduction of Lab 27, pause the video while you do so, and then return when you are ready to start on the materials and methods, and be ready to flip forward and record results as they are shown in this video. Again, if this goes too fast, you can always pause or rewind. So, the list on the left are going to be the bacteria that will be involved somehow in this lab. You'll notice in the methods section, there's a lot of assays that are performed as part of this lab. Now, not all of the species that are listed are going to be performed in every assay, but each species on this list is going to be involved in at least one assay. Now, a feature that is interesting is that most pathogenic cocci tend to be gram-positive rather than gram-negative. So let's take a look at the gram stain results for some of these species. Again, please record the results in your lab. You should be recording the gram stain, the shape, and the arrangement, such as Staphylo, Strepto, or Diplo. So one of the things this image demonstrates is that the streptococci do not always show a strepto arrangement. So interestingly enough, many of the species that are taxonomically identified as belonging to the genus Streptococcus do not actually show a strepto arrangement under the microscope, but rather are grouped in with the Streptococcus genus because their DNA indicates that they're evolutionarily related to other more prominent Streptococcus species such as Streptococcus pyogenes. So what this really shows is that it is not a good idea to rely strictly on arrangement, such as looking for a Streptococcus arrangement under a microscope, to help you determine if the unknown species that you have truly belongs to Streptococcus or belongs to some other genus. Again, make sure you record the results in Lab 27. For Escherichia coli, you will need to look up the results of your gram stain from Lab 10. So now that we've covered the gram stain, let's take a look at Part B, which is inoculating 4 species on mannitol salt agar. Now, mannitol salt agar has a lot of salt in it, and that salt makes this type of media selective, in that it selects only for salt-tolerant species. The lab asks you to divide up your agar plate into quadrants, and then inoculate each quadrant with the species shown. So let's assume that we did that. 48 hours later, here are our results. So you'll notice right away that E. coli has no growth, and that is because the salt inhibits E. coli. Where Staphylococcus aureus and Staphylococcus epidermidis, both salt-tolerant species, grow just fine. And in our laboratory, Micrococcus luteus tends to show pretty good growth as well. Now, in addition to the growth, you'll notice that Staphylococcus aureus has turned the agar yellow, while the other species keep the agar pink. In fact, that difference between Staphylococcus epidermidis and Staphylococcus aureus is one of the easiest ways to distinguish between the Staphylococcus epidermidis, which is found on your skin but rarely pathogenic, from the much more pathogenic Staphylococcus aureus. So a positive result for fermentation or mannitol fermentation is turning the agar yellow, and you see that clearly here with Staphylococcus aureus. Alright, Part C of the lab asks you to inoculate blood agar. So blood agar is going to basically be a general purpose media that is infused with whole red blood cells. And because the whole red blood cells are suspended in the agar, it actually makes the agar opaque and not transparent. So we would inoculate blood agar with various Streptococcus species, and different species show different hemolysis patterns. So if a species completely lyses the red blood cells, we call that beta hemolysis. If it shows partial lysing, and partial should be in quotes because there is no such thing as a partial lysing, but it does seem to lyse a little bit and then ends up changing the color of the hemoglobin to make the blood agar look more yellowish. We call that alpha hemolysis. And if it doesn't really do much to the blood at all, we call that gamma hemolysis. So here are the hemolysis patterns. So you can see over here that we have complete clearing, and if I were to hold up my fingers behind the plate, you would be able to see them pretty clearly because the red blood cells have been completely lysed in the area that the bacteria are growing, and therefore the agar becomes transparent. This is called alpha hemolysis. You can see the yellow hue around the bacteria, but clearly it's not as clear as it would be for beta. So again, we call this partial hemolysis, and that really should be in quotes. Gamma hemolysis might show a small amount of yellowing, but largely leaves the red blood cells untouched. So why do we do this experiment? Hemolysis patterns are really useful in helping us distinguish between the various Streptococcus bacteria. So Streptococcus pyogenes, which causes strep throat, flesh-eating disease, it can cause toxic shock syndrome, it's really pathogenic, shows beta hemolysis. Streptococcus pneumonia shows alpha hemolysis, and another streptococcus that we looked at in a previous lab, Streptococcus mutans, which is found in the mouth, also shows alpha hemolysis patterns. Typically, non-pathogenic Streptococcus species show a gamma hemolysis pattern, which is to say no hemolysis at all. So again, if it's non-pathogenic, it tends to be gamma. Part of this lab asked you to do an unknown with a simulated throat culture. Obviously, that's not going to be done here, so you can ignore that part of the lab. Okay. If we get a Streptococcus species, so it belongs to the genus Streptococcus, and it is beta hemolytic, there's actually a number of potential microbes that it could be, how can you tell the difference between Streptococcus pyogenes and other beta hemolytic species? Likewise, if you have a Streptococcus species and it's alpha hemolytic, how can you tell the difference between Streptococcus pneumoniae and other alpha hemolytic species, of which there are many? So this flow chart here shows you what you could do. So if you see here, we have a Streptococcus species, and then if you grow it on blood agar and it's beta hemolytic, then your next step is to culture that species in the presence of a bacitracin disc. Bacitracin is a type of antibiotic. If it's alpha hemolytic, then you culture that species in the presence of optocian discs, which is another type of antibiotic. So the reason is because Streptococcus pyogenes is sensitive to bacitracin, where other potential beta hemolytic streptococci can grow right up to that disc and are resistant to it. Likewise, Streptococcus pneumoniae is sensitive to optocian, whereas other alpha hemolytic streptococci grow right up to that disc because they are resistant to optocian. So this image here shows Streptococcus pyogenes cultured in the presence of a bacitracin disc. So this is a disc infused with bacitracin. You can see that the bacitracin has clearly leached out into the agar, and there is that zone of inhibition where Streptococcus pyogenes cannot grow because clearly bacitracin kills it, where Streptococcus pyogenes is growing in this other area. And you can see that clearly because you can see the beta hemolysis. The aptotion disc, let's take a look at two different species of Streptococcus, one of which is sensitive to aptotion. That's going to be Streptococcus pneumoniae, and again you can see that zone of clearing there. And whereas most other forms of Streptococcus are resistant, and therefore the disc does nothing to inhibit their growth. So please record those results in your lab. Alright, the next experiment is called the bile esculin test. So the bile esculin test is used to help distinguish between Enterococcus faecalis from other gram-positive Streptococcus. Now, as a general rule, Streptococcus tends to be very sensitive to salts, meaning that high salt concentrations are going to inhibit Streptococcus species from growing. Enterococcus faecalis, which was once grouped with the Streptococcus, actually, and is sometimes still called Streptococcus faecalis, is unique. The term entero means gut, and because there's a lot of bile salts and other types of salt in the gut, Enterococcus faecalis actually can tolerate salt really well, including bile salts. So if we inoculate Enterococcus faecalis into something that has a lot of bile in it, it should still be able to grow, whereas most other gram-positive Streptococci do not grow. So Enterococcus faecalis, when inoculated in bile with a substrate known as esculin, will actually be able to grow, metabolize the esculin, and then as part of the chemical reaction that happens in the test tube, forms a dark brown color. Whereas species that are unable to grow or do not grow well in the presence of bile do not change the color of the test tube as shown here. So this lab would have had you inoculate Enterococcus faecalis and another Streptococcus species, Streptococcus lactis, and these are the results you would have gotten. So Enterococcus faecalis shows that dark brown color, indicating that it was able to grow in the presence of bile salts and metabolize the substrate to produce this dark brown color, whereas Streptococcus lactis either grew poorly or did not grow at all. Okay, so why do we do the bile esculin test? Prior to more sophisticated DNA technology, we were separating out the Streptococcus species based on their surface antigens, and they were designated by letters. So there would be type A Streptococci, and type B, and type C, and type D. Well, Enterococcus faecalis represents the group D. So it turns out that group D strep, again, Enterococcus faecalis used to be called Streptococcus faecalis. So Enterococcus faecalis is a type of group D Streptococcus, and it turns out that group D tests positive for bile esculin, where almost every other type tests negative. So the bile esculin test was used to separate out the species that would be categorized as group D Streptococci, including Enterococcus faecalis. Alright, I mentioned that Enterococcus faecalis likes salt or tolerates salt much better than most Streptococci do. So another way that you can test for salt tolerance is doing a salt broth test. This is using a typical nutrient broth, but it's using a 6.5% salt. You inoculate your species. If it grows, the broth is turbid or cloudy. If it doesn't grow, the broth is going to be clear, just like the control. So Enterococcus faecalis shows the cloudy or the positive test, again indicating that it grows quite well in salt. Streptococcus lactis does not tolerate salt, does not grow, and gives a negative test. Alright, the next test looks at something called coagulase. So your instructor may have discussed coagulase as a virulence factor that is associated with pathogenic Staphylococci. So in Staphylococcus, there's two major species, Staphylococcus aureus, which is considered to be the pathogenic staph, and Staphylococcus epidermidis, which is found on everybody's skin, and it can cause disease, but it's really unusual. So coagulase is a major virulence factor of Staphylococcus aureus. So one way that you can distinguish between Staphylococcus aureus and Staphylococcus epidermidis is to inoculate your species of Staphylococcus into a test tube with serum. Now serum is going to be plasma that has been defibrinated, or the clotting agents have been removed. And then what happens is, if the species produces coagulase, it ends up creating a clot, like kind of a chunky jello-like substance in the plasma. So in the lab, what we would have done is taken some serum, and we would have inoculated Staphylococcus aureus in one and Staphylococcus epidermidis in another test tube. And what we're looking for is the chunky clot feature that tells us that that species is coagulase positive. You'll notice that the test tubes are slanted. The reason why they are slanted is to show whether or not the fluid inside is liquid or more solid. So you'll notice that the original serum is liquid because when you turn the test tube on its side, the liquid obviously moves. When you inoculate that serum with Staphylococcus aureus, it creates a chunk, a clot. So you can see that clearly with Staphylococcus aureus, whereas Staphylococcus epidermidis does not produce coagulase. The serum remains liquid, and you can see that in this image. All right, next up is the oxidase test. The oxidase test is run on a strip where what you do is you add a little bit of bacteria to the strip. If they possess an enzyme known as oxidase in reasonably high quantities, then they will turn a substrate that's on the test into a product that is colored, and that colored product is kind of a royal bluish purple color, as you can see here. So the test strips are pretty small, but typically what you would do is you would run a variety of species on one test, and so we're going to mimic that here. So a positive test is going to be purple, a negative test is yellowish. So Streptococcus species show kind of a yellowish color. Staphylococcus aureus, so again, record your results. Staphylococcus epidermidis. Oh, Neisseria. So Neisseria shows our first kind of purplish bluish color, indicating a positive oxidase test. And then the last one is Micrococcus. Micrococcus is negative. So the last test that we do is the Catalase test. Now the Catalase test is looking for the presence or absence of the enzyme catalase. What catalase does is it takes hydrogen peroxide and breaks it down into water and oxygen gas. And it's really useful to aerobes to have this enzyme because hydrogen peroxide can build up around the ATP making machinery, and hydrogen peroxide, as you may have learned, is toxic to most microbes. So they want a way to kind of quickly break down any hydrogen peroxide that might form into the much less toxic water and oxygen gas. So if you've ever had a wound and you put hydrogen peroxide in it, you might see bubbling. And that bubbling is from your own catalase producing water, but also oxygen gas that produces bubbles. So we can apply that to bacteria. We can take a microscope slide and throw a whole bunch of bacteria on it, and then drop hydrogen peroxide on top of the bacteria. And if they are catalase positive, then they should form oxygen bubbles, or there should be some bubbling on the microscope slide. But if they don't have catalase, then they are considered to be negative. So you can see that here, where you've got the bubbling on the left, but the no bubbling on the right. So when we test this against our various species, only Streptococcus species are negative for catalase, whereas everything else is positive for catalase. Now, one of the other things that we didn't get to really do in this class is when we grow up Streptococcus species on blood agar, we put them in what is known as a candle jar, and that is shown in the left image. A candle jar is one where you put the cultures in this big jar and you light a candle and then you seal it. And the candle flame burns the oxygen until it basically consumes most of the oxygen and the candle goes out. And what that does is decrease the concentrations of oxygen in the jar. It turns out that most Streptococcus species grow best when the oxygen concentrations are lower. Not zero, but lower. And this actually relates to the Catalase test, because Catalase gives the benefit of taking any toxic hydrogen peroxide and breaking it down into water and O2. Well, it turns out that most species that have difficulty with high concentrations of oxygen do not make catalase, because this reaction that you see here is reversible. In other words, water and O2 gas can combine and form hydrogen peroxide. So by having the enzyme catalase available, if you're a microbe, you can quickly break down that toxic hydrogen peroxide back into its water and oxygen gas. But if you're a Streptococcus species, you don't have this catalase. So if any hydrogen peroxide builds up, then you really don't have any way to deal with it, and it's incredibly toxic to you. So by reducing the concentration of oxygen in the candle jar, less hydrogen peroxide is produced, and therefore the bacteria grow a little bit better. Now this concludes Lab 27. However, I'm going to continue on because your instructor may assign some additional assignments that have to do with creating a table and/or reasoning key. So please check with your instructor to see if they're going to do the supplementary assignment. So we went through a whole bunch of different tests. And one of the things that should be clear is that these tests can be used to differentiate between closely related species. So one way to basically come up with a system to help you identify some unknown bacteria is to create what is known as a reasoning key. So let's say that I give you an unknown culture of a bacteria and it happens to be one of these species, and your goal is to identify it. How would you do that? Well, what you would need to do is you would need to basically separate out these bacteria by their experimental results. So to start, you make a table of all the species and all the experimental tests that we did in this lab, and you basically record the results. So I've gone ahead and started it for you. Again, if your instructor is prescribing this, you need to pause the video, you need to copy this table down, and you need to use the experimental results covered in this tutorial and fill out this table. Once this table is complete, your instructor may follow up with the class and show you how to use this information to create a reasoning key.